go. Hello, everyone. Welcome to workshop number 4B on advanced data analysis. This is part 2 of our series on data analysis. Part 1 was 2 weeks ago, covering more basic ideas. So we hope today's workshop can help you gain more in-depth ideas on most crucial component of your science fair project, data analysis. So once again, this presentation will be recorded. So please mute your mic and turn off your video during the workshop. If you have a question during the presentation, please type it in the chat. And after today's presentation, a copy of the slideshow and a video of today's presentation will be available on the gsdsef.org website. We'll go through some brief SLB introductions. I'll go first. My name is Stephen. I'm a sophomore, and this is going to be my 4th year competing in the fair. I'll hand it off to Alex.
Hello, my name is Alexander. This is going to be my 3rd year competing, and yeah.
All right, thank you. And we also have Mr. Rodeker in attendance today. He's the fair director. If you have any questions that you put in the chat, I'm sure he will be more than happy to help with those. All right, just some PSAs ahead of time. Our next workshop will be on January 10th in the new year. This will be on screening, digital project slide deck, and your project notebook. So please keep in mind that your SRC review, this is the clearance that you get to start your project, that is due on December 31st. If you haven't done it already, we highly encourage you to get it done with right away. And please don't wait until the last minute on December 31st to do it, because SRC review is only clearance for you to start your project. So if you submit your project to SRC, not like on the last minute, you can only start on January 1st. So after the SRC review, the next big deadline is for the quality screening. So this is just how the screening committee can verify if you are using proper scientific practices and make sure that you are doing your project the correct and scientific way. So you can see these deadlines on the screen right now. And you can also find these deadlines on the website. And as always, you can follow us on Instagram for updates and new workshops, and our YouTube channel for workshop replays and tutorials for things like the SRC review and the quality screening process. All right, today's agenda will first have a recap of what exactly is data analysis, and then we'll go over various types of analyses and when to use them, like error bars, box and whiskers, statistical tests, confusion matrices, etc. Don't worry if these sound unfamiliar to you. For this workshop, we'll help break it down, and then if you want to use it for your project, you're more than welcome to go on your own and do some more research that way. All right. First, I'd like to go over analyzing the data first. So make sure to organize your data in charts and then graph them in order to see the correlations. You should first observe your graphs and then calculate some statistical results, and then discuss the differences and the meaning of your statistical analysis, and then relate it back to your problem statement and hypothesis. Ultimately, your data should be answering a question about a problem and not supporting or disproving a hypothesis. And just a note, sometimes you can be fooled by looking only at raw data and make an incorrect assumption that the data does or does not support your hypothesis. So please carefully follow the steps for graphing and interpretation of your data, or you may miss important info and new ideas suggested by your data. And then some more reminders, about organization. Please remember to organize all your numerical data in tables. We recommend software like Excel and Google Sheets. And qualitative data, such as photos and observations, should also be kept neatly. All of this can go in your appendix, which is the section in your FAIR notebook for your project's raw data. So you're going to want to keep it nice and neat so that on judging day, all your judges can walk by, take one good look at your appendix, and then walk away with a good sense of what you did to collect data. And perhaps most importantly, make sure to save all your data, even pre-trials, and even if you think it's useless. All this data is valuable in proving a point, small or not, and you may want it later or come back to it and make some interesting suggestions in your discussion for future work or reasoning behind your experiment. And just a reminder, all of your data goes into the appendix. So regardless of what trials that went wrong, or perhaps some that you don't like as much, all that goes into the appendix, just so we can get the best idea of your project. All right, Alex, I'll let you take it from here. Alex, are you there?
Am I muted?
Not
Okay, sorry. So now we're going to talk about what types of analysis you'll most likely be using. So not everything that we present today is going to be relevant to your specific project. These are just the types that anyone um, can apply to some people. So Um, we recommend that you depend on your literature. Like when you did your review of literature, go back to that and try and use similar types of analysis or the same ones as similar projects in your field. In addition, like later we will have a questions portion, and if you want to ask us questions about what type of analysis you should use, then we'll be happy to answer. And remember, don't just plug and chug pretty much. Science is about understanding the math and understanding your analysis. So don't just randomly put in numbers, try and see if they have any connections and what it really means in the context of your experiment. Stephen? Okay, so every data point has some error. So when you're generating and plotting data, you're either plotting the points you measured directly or you're plotting a mean value for different conditions. And remember, mean is just the average. The data points you directly plot are always associated with an error, and depending on the machinery or method you use, you may not know the value of your error. For example, a standard ruler only has markings to a millimeter, and if you're measuring in centimeters, your error would be half the precision, which would be half a millimeter, or 0.05 centimeters. When plotting this, your error bars would be 0.05 centimeters above and below your measurement. On the other hand, if you're plotting averages and you often want to know to show the distribution of points you averaged, so you know how precise your data is or how well the points agree on a value. For example, you could use a standard deviation, which is a mathematical formula. If you want to learn more, you can Google it. And you can calculate it in either Excel or Sheets or by hand. And all of your points in that condition and plot is it as an error bar, and with your average, you can know if your average represents your data accurately and if its distribution overlaps with any other conditions. Okay, so now moving on to box and whisker plots. In addition to error bars for your average values, another way to show data distribution is with box and whisker plots, which pretty much is a 5-value summary of your data. It shows your minimum and your maximum value. For example, on this graph, you can see on the far left, it has a minimum, and on the far right, it has a maximum. The length and sorry, it also shows where the 25 and 75% of marks of your data are, which are the blue and yellow lines in the top box. and everything in between that is anything between 25 and 75%. Um, the length and distribution of your plots can tell where most of your data points are, and if the highest and lowest values are outliers, which don't represent your data. For example, in the second box, it is relative. The box is relatively smaller than the total length and is placed towards the lower value, which is where the majority of the data points are, because there's not many points that are higher. So if you're looking at the second box, yeah. Okay. Um, Now let's say you have one variable that differs over time and it's in a scatter plot. For example, plant growth over 1 week. You can ask if there's linear relationship between plant growth and time, and perhaps you want to know how tall a plant will be in 2 weeks. You can use a line of best fit to calculate the slope, and therefore you can calculate how tall it would be after 2 or 3 weeks. In this case, you would ask, if X variable, which is the days, increases, does the Y variable also increase? And the Y variable here is plant growth. And if this is a positive correlation, which pretty much means the line is going up, then you, as designated, the positive slope of the line. On the other hand, it could also be going down, and you'd do the same, um, and you'd still find the height after 2 or 3 weeks. The intuition behind the line of best fit is pretty much basically you are trying to minimize the distance from each data point to the predicted line. You're trying to make the line that best fits your data points. Later, we'll talk about how to use a statistical test to quantify how well the line of best fit represents your data. Okay, so our statistical test we have is R squared, also known as the coefficient of determination. It's a value that tells you how well your model or line represents the variance of your data. Basically, how well does your line fit the scatter plot? A higher R squared means a higher R squared means a better fit with less error or deviations from the line. In the figure, you can see that an R squared of 100% or 1 is perfectly on the line. In the top left one, it's perfectly on the line, so that would have a coefficient of determination of 100%. If you use linear regression or any type of regression, such as fitting exponential graphs or logarithmic graphs, you should use an R squared value to validate and quantify how well your model is doing, and if you can really make a statement about the correlation between the independent and dependent variable. Yeah.
So next we're going to be talking about statistical tests. Next slide. So beyond just graphing, sometimes you actually have you might have doubts about how accurate your data really is, and this is where you would use statistical tests, which are basically like a series of different tests for different like reasons that you would test how accurate your data is. Uh, Keep in mind that in reality, most experiments usually have very large populations, which are the total amount of data points that are in existence. And going back to plant growth, if you were doing a project on growing radish seeds that we were talking about in the first few workshops, the actual populations that you're trying to understand is the total amount of radish seeds in existence, which is impossible for you to measure. Therefore, you have to take representative samples, which is like a random subset that we assume is representative of the population, and you can measure the within the scope of the experiment. To further convince others of your result, you can show that the probability of your result being totally random is very small, using all these statistical tests that we're going to be talking about today. There's a lot more than what we're just going to be talking about today, based on your type of project, you should do further research in that. But these are the primary types. Next slide. So This is some like simple vocabulary of what you're gonna be seeing very commonly for statistical tests. So first, we have to understand the basis behind these tests. In every project, there's a null hypothesis where the default hypothesis is that the null hypothesis is the default hypothesis that you're trying to disprove. It's basically saying that variable X is not related to variable Y, and that the results we get if they seem to say otherwise are basically due to chance and they are insignificant. The opposite of that is the alternative hypothesis, which is usually the if-then hypothesis we encourage to learn first. It says that variable x does affect variable y, and for example, if we're talking about growing radish seeds under different pre-treatments, then the null hypothesis would be pre-treating radish seeds that doesn't affect plant height, and the alternative would be pre-treating radish seeds that does affect plant height. So the p-value is the probability value that basically determines whether you can disprove or reject the null hypothesis and accept your alternative hypothesis. It's basically the probability of your null hypothesis actually being true. The probability of your results being completely or the probability of your results being complete coincidence. The standard is usually to have a 95% significance threshold, which means that for your results to be considered statistically significant, your p-value cannot exceed the critical value of 0.05 or 5%. Next slide. So the t-test is a common method for like comparing two different populations with for example, like a control and a variable group. It's what produces a p-value, which we just talked about in the previous slide, and you could use that to determine how likely it is for these populations to actually come from the same conditions and randomly end up in this configuration. Or in other words, you could see how significant their differences are using the t-test. It's used when you have numerical data that's defined in two different categories, such as the plant height of the radish seeds versus the carrot seeds. And you can calculate this in Excel and Google Sheets. And the widely accepted critical value for the rejection of the null hypothesis is 0.05 or 5%. So, for example, in this plot, the p-value from the test exceeds 0.05, and therefore it is not grounds to reject the null hypothesis, meaning the data did not statistically support the alternate hypothesis. Basically, this shows that the results aren't statistically significant between the control and variable groups. So the t-test is very, very important to find that out. Next slide. Okay. The z-test is basically a test that converts the difference you observe into a z-score, and this test is primarily used when you have a population, you have a standard deviation of the population, and the data is normally distributed like in this bell-shaped curve that you see over there. That's when you would use a z-test, and basically, the z-test overall, it converts the difference that you observe into a z-score, which then tells you how many standard deviations it is from what you would expect. The result is interpreted using the standard normal distribution to get a p-value. If the p-value is small, the difference is unlikely to be due to chance alone, and z-tests work best with larger sample size. So, for example, if the yeah. So if the p-value is small, it's basically like it's not because of chance, and you could also use a t-test for that, but this is specifically for population standard deviation if it's known. Um, Z-tests work best with large sample sizes, even if the data isn't perfectly normal, and they are often used when results need to be compared against a very clear benchmark or standard. Next slide. So the cheese squared test is a complete statistical test. Um, and it's basically commonly used for testing genotype data and predictions for inheritance of certain traits. It's for testing purely categorical data, such as comparing different body colors or wing shapes. And this is a very specific test that's always used in genomics, but be sure to look up if you actually think it applies to your work. It's interpreted in a similar way to the student's t-test with a critical value. However, this value does change depending on how many categories you're testing, so be aware of that. Next slide. So this is a confusion matrix. Confusion matrix are very, very commonly used in machine learning projects and classification projects. And it's a very useful metric to actually show how accurate your model is. Um, ba Basically, here you could get details on how well your model is predicting, specifically how often it's predicting true versus false. For example, if we were building a model to predict the plant type based on height, where we have a height threshold of 3 inches, um, and with the known information that any height below 3 inches is the radish plant, we could use a confusion matrix. Here we can know how often can our model actually predict if a plant is a radish plant or if a plant isn't a radish plant. There are true positives and negatives, and on the other hand, how often does our model predict incorrectly, where it predicts that an actual radish plant isn't a radish plant and vice versa? These are the false positives and false negatives. There are additional useful metrics associated with this as well, such as sensitivity, specificity, accuracy, and precision matrices. They're usually paired up with confusion matrices and ROC curves, which we're going to be talking about in the next slide. But you should definitely conduct some research and um, see what best applies for your project. Next slide. So ROC curves are very, very important statistical tests, similar to the confusion matrix for machine learning models. An ROC curve or a receiver operating characteristic curve is basically it's directly related to the confusion matrix, and it basically like the curve shows how good a model is at telling the difference between two things like positive and negative as you change its sensitivity, or the true positive rate, which is represented by the y-axis. Um, Going back to our example, note that we from the previous slide, note that we actually set a threshold randomly at 3 inches. However, a lot of the time we don't know the best threshold. So that's what an ROC curve does. It basically reports the true positive rate and the false positive rate for a series of thresholds and shows a curve for the data. The closer the curve is to the top left corner, the better the model. A random guess is a diagonal line, and a perfect model hits the top left corner. We won't go into the specifics. However, if you're doing a machine learning project, be sure to contact one of today's presenters. And also another thing with ROC curves is that if it shows like a very, very like good looking ROC curve that kind of like fits the criteria that we just talked about in every way. And it's very, very like it's perfect 100%. Make sure you actually look through your data and how you train the model, because that often ends up being like a problem with underfitting or overfitting.
All right, that wraps up workshop number 4B. Once again, thank you for coming. Our future workshops are displayed on screen. The next one will be January 10th on the screening process. As always, if you have questions, please email gsdscfslb@gmail.com. And I will send out the link for the mentorship program one more time. And this concludes the recording.